This conference will now be recorded.
A very good evening to one and all present here. I'm Ms. Neha Mahajan, Executive Organizer. On behalf of Biosetup Lifesciences, take this honor to welcome our Founder Coordinator, Argedeep Das Sir, Event Coordinator, Dr. Arpana Sharma, Dr. Ritu Chandil, Dr. Sudeep Das, and our today's eminent guest speaker, Dr. Laura Harris, faculty members, organizers, and all my dear student participants. I welcome you all for the international webinar on the topic Gene Expression and Pathway Activity Meta-Analysis of SARS-CoV-2 Infection in Lungs, organized by Biosetup Lifescience in collaboration with Michigan State University and ICER. I hope you all are good and safe at your end. Before starting the session, a humble request to all the participants to please do not share your audio, video, or screen sharing in order to avoid any kind of disturbance. If you have any doubt during the session, please use the chat box. Filling of the feedback form is mandatory for your attendance and certificates. Now let me tell you about Biosetup Lifescience. Biosetup Lifescience is a hub of scientific research and learning approved by ISO 9001-2015 certified organization and also MSME registered under Government of India. We have multidisciplinary team of eminent and experienced faculty members and research associates that provide support to aspiring students in life sciences. We have successfully organized many webinars, workshops, internships, e-training programs, certification courses, career counseling, and competitive exam preparation. That is for CSIR, IIT-JAM, JNUee, IISc, and DBT. We are continuously working for the career improvement of students. As we all are aware, we have been facing difficult times during the ongoing pandemic since 2 years, but we believe in not stopping and giving quality education to the budding minds through online platform. You must have also heard that empty rake makes more noise. So it's important to keep filling the bucket of knowledge through any source available. Keeping this in mind, I would like to introduce our event coordinator, Dr. Chandil and Dr. Das. About Dr. Ritu Chandil, ma'am, she is MBBS, MD in biochemistry, as well as a very renowned speaker and tutor. Ma'am is currently working with Bridge Candy Hospital Trust, Mumbai, Maharashtra, and she has been published multiple articles in various national and international journals and academic books. Next, about Dr. Sudeep Das is a lung infection biologist and human microbiome expert. Currently, Sir is postdoctoral fellow, University of Lausanne, Switzerland. He has received several accolades and high-impact publications in renowned international journals. He was a Swiss Academy of Science Scholar, DAD Fellow, DSG Career Development Fellow, and currently a holder of highly prestigious Marie Curie Fellowship from the European Union. Now I would request Dr. Chandil to guide us with her golden words and introduce our today's eminent speaker, Dr. Harris. Over to you, ma'am. Thank you.
Good evening, everybody. Let's begin this academic feast, and I would like to introduce Dr. Laura Harris, ma'am. Dr. Laura Harris is the Director of Training for the Institute for Cyber-Enabled Research at Michigan State University. She is an educator who enjoys developing curriculum that involves use of computational resources and also enjoys conducting and mentoring others in data science and biomedical research. Her projects use bioinformatics, basic laboratory approaches to examine differential gene expression changes associated with antibiotic resistance in a variety of bacterial species, microbial-induced cancer, and more recently, SARS-CoV-2. Dr. Harris is in collaboration with the American Society for Microbiology and has co-authored several educational and scientific papers. She also has received several honors for her leadership excellence, including Lead Like a Woman by the Michigan Chamber of Commerce and honorary membership to the National Society of Leadership and Success, Sigma Alpha Pi. It's our privilege to have you here. Now, may I please request you to kindly proceed. Dr. Harris.
Yes, thank you for the wonderful introduction. Am I good to go? Hopefully you can hear me, yes?
Yes.
Can hear you.
Yes,
Oh,
yes, ma'am, you're audible.
Good. Good evening. As you can see from my background, it is still morning here, and I am looking forward to my second cup of coffee later today. But I'm really excited to talk to you about the SARS research that we've been doing at Michigan State University here in Michigan in the United States of America. So as my talk is titled, Gene Expression of Pathway Activity Meta-Analysis of SARS Infection in Lungs. We will be looking at SARS-CoV-2, which is the causative agent for the COVID-19 pandemic. We will also be looking at SARS-CoV-1 and MERS. So let's get started. So my talk today will include a brief background and introduction to not only the COVID pandemic, which we are all familiar with, but also an introduction to my project. And my project has three distinct aims. So the first aim is to identify gene expression and pathway activity changes associated with SARS-CoV-1 infection. And I'm primarily looking at human primary tracheobronchial epithelial, basically lung cell cultures. Once I've identified genes that change expression in response to a SARS-1 infection and the pathways that change activity inside a human lung cell as a result of a SARS-1 infection, my second aim is to verify the gene and pathway findings from aim 1 using similar human lung cultures and random modeling. Once I'm firmly convinced, or at least I'm, comfortable, that the genes and pathways that I've identified through aim 1 and verified in aim 2 are really associated with SARS-CoV-1, then I want to look across not only other models, such as in vivo mouse lung samples, but also other SARS strains, as I mentioned, SARS-CoV-2 and MERS. And then I'll give a brief introduction and some further considerations. So I started as a microbiologist back around 2000. And most of my training in the last 20 years and work has been in microbiology or some sort of inflammatory disease. So I'm well versed in the biology. In the last 5 years, I got my PhD in biomedical informatics, looking at computational ways to approach antibiotic resistance in bacteria. When we had the pandemic breakout last year, all of the work I had done towards bacteria got shuffled into viruses, and here we are. So when I started as a microbiologist, the beta coronavirus family was nothing new. It is the causative agent of the common cold, which is usually non-life-threatening. It's annoying for 7, maybe 14 days. You cough, you sneeze, you have runny eyes, you're a little miserable, but you get over it. It wasn't until around 2003 when we had SARS-CoV-1 emerge, and that then caused severe acute respiratory syndrome, aka SARS. So fortunately, SARS was contained and it didn't become a widespread pandemic. A few years later in Saudi Arabia, the Middle East respiratory syndrome, or MERS, came out, and that then was related to SARS-CoV-1. Again, contained, did not become a pandemic, life went on. Then, as we all know too well, SARS-CoV-2 came around last year. It is the causative agent of COVID-19 and why I'm not in India presenting this to you in person, why I'm still on my porch here. So I hope you all are well and your family as well. But as you know, there's limited treatment options for all SARS infections, including COVID-19. And fortunately, there are vaccines available, but right now we're struggling to get those vaccines out to the remote areas. And so India, for example, struggles quite a bit in getting vaccines to its remote regions. And so COVID-19 is still a raging global pandemic. So my overall goal with my project is to find molecular changes inside lung cells that change in association with a SARS infection. And I don't have a lab that can do hardcore SARS bench research, so I have to use a computer. So I'm going to computationally analyze mRNA expression data, which I'll express in a minute. And this is to identify potential gene and pathway targets that maybe we can use to develop therapies to treat COVID-19 patients. So my project is the first time that we've compared gene or pathway signatures, which I'll define in a minute, in order to identify genes and pathways associated with SARS infection. Further, it's the first time a meta-analysis regarding genes and pathways has been done. We looked at over, 12 data sets containing 37 signatures, which had, I want to say 9 different strains. And then we also included the mouse model in addition to the Petri dish of human cells. So this is the first time that computational analysis has been done. So I'm going to assume most of you have no master's degree in biology. I have 2 actually. So let's start basic. So a gene is a piece of DNA that is the basic physical and, more importantly, functional unit of heredity. So genes are stored in your DNA. Humans have around 30,000 different genes, and you will send copies of the genes to your children. If a cell wants to express a gene, in other words, turn it into a protein product that's used to build the cell, make the cell run, that sort of thing, the cell will make a copy of the gene from the DNA, and that copy is a transcription into a messenger or mRNA. This mRNA will usually leave the nucleus, it goes through translation, and it's the copy that becomes proteins. Proteins are the building blocks of your cell. So this process is called mRNA or gene expression. Okay, and that's what we're looking at, is which genes are effectively being made into proteins. So how do we do that? Well, in order to get gene expression data, we're going to take sample of RNA from one condition, say SARS infected sample, and we're going to take RNA from a second group, say mock or uninfected sample, and we're going to compare the mRNA that binds to specific nucleotide probes. Nucleotides are sequences, A, T, C, G, for example. And from the picture that we can get, the computer can measure different colors and fluorescence and binding, and you effectively get what you see here on the right side, a list of genes, and then you have samples with numbers, and these numbers indicate intensity of color. The higher the number, the more expressed that gene is in that sample. I'm going to stop for just a second and check the chat and make sure I have no questions. I'll do this periodically because I know some of this gets detailed. All right, if you have questions, plop them in the chat. Yep, DNA is boss, RNA is manager, proteins employee. Sweet, I feel confident then. Let's move on. Oh, my apologies. My idiot dog is going off in the background. Fun from working home. All right, so let's discuss what a gene signature is. So you just saw this mRNA expression data, and I've clearly defined my SARS infected samples and my mock infected samples. So I can use a statistical metric like a t-score from a t-test. And that can tell me the general expression difference between my SARS infected group and my mock infected group. And I can then tell by the t-score if a gene went up, it would be a positive t-score, and I'm going to color that red. And if my gene goes down in infected versus uninfected, it's going to be a negative t-score, and so that's going to be colored blue. Genes that don't have much of a change are going to be around the middle of my signature, and that's going to be around white color. Okay. So a gene signature is a list of genes ranked on their differential expression, how much the gene expression changes between SARS and mock infected, as defined by, in this case, a t-score. So this is just another way to look at it. So my red genes, these are overexpressed in SARS versus mock. They're going to be at my positive tail or the red end. If I have underexpressed genes in mock compared to in infected versus mock, it's going to be blue, and that's my negative tail. And I see my no change in the middle. So if I really want to look at biological relevance, I care about the genes in the positive tail and the ones in the negative tail, because those are the genes that are most differentially expressed. Pathway signatures are similar in concept. So I'm going to take the gene signatures I just made on the prior slide, and I'm going to compare them to around 7,500 individual gene sets from gene ontology. And I'm doing this using gene set enrichment analysis, which I'll describe in detail in a minute. So this gives me 7,500-ish pathways that then have a score based on gene set enrichment analysis. And I can use that score to rank the pathways based on their activity. Same idea as before, the red pathways would be upregulated in SARS compared to mock. The pathways in the blue end would be downregulated, negative normalized enrichment score, and they'd be at the blue end. And pathways with no real change between SARS and mock would be towards the middle. And again, I care more about the positive end and the negative tail than I do the middle. Okay, so these are kind of fundamental. I'm going to take a moment, just check the chat. Do I have any questions? Are we good? Okay, so I do have a question. How do you get data for specific gene expression? Is it done wet lab or just predicted computationally? Very good question. So I'll explain in a second that all of this was generated in the wet lab by other labs. So when a wet lab does publication of their data, they're required in the United States to upload it to a government repository that's called the Gene Expression Omnibus, or GEO for short. And so I have access to all the lab data that's generated in my country, and then I can use it for computational analysis without ever stepping foot in the lab. Does that answer your question, sir? Awesome question. Any other questions before I move on? Okay, feel free to plot more in there. I will check back in a bit. So my first aim is to identify gene expression changes and pathway activity changes associated with SARS-1 infection. And the reason I'm picking SARS-1 infection is because when we started this project about this time last year, very little data was available on SARS-2. So that part had to be added on later. So we're starting with SARS-1, the one back in 2003. So I was lucky that I found in the GEO a super series where 3 data sets were made exactly the same way by the exact same people. So I didn't have to worry about things like differences in cell types, or differences in when things were treated or harvested, or differences in the process of collecting the samples for samples for analysis. It was consistent, and that helped me identify my genes and pathways. So I have GSE 47960. The number's not important, but if you want to look it up in GEO, that's how you find the data. And so from this data set, I have 4 samples that are infected with infectious clone. That's what IC stands for, SARS. So this is a fully virulent SARS-1 strain. And then I have a few samples that are mock, and I'm going to make my gene signature just like we described before. I'm going to take a second data set that also has iCSARS and mock samples, and I'm going to make a second non-overlapping signature. I'm going to compare these signatures using gene set enrichment analysis. But to do that, I have to define my own query sets. So I do that by taking one signature, and I take the top 500 genes and the bottom 500 genes, and these become my query sets. I then compare each query set to the entire reference signature, even the genes in the middle that show no change. This is an advantage of using gene set enrichment analysis over something like a Fisher's exact test, if you're familiar with that, for example, from enrichment analysis. So the top panel here is my enrichment plot for the positive 500 end, and the one on the bottom here is the enrichment plot for the negative 500 end. Okay, and so what you'll see is you see at the bottom the reference signature. On top of it, you'll see these lines. The lines indicate matches between the genes in my query and the genes in my reference signature. And what you'll notice is that the genes in the positive end tend to cluster towards the red end of the reference signature, whereas the genes from the negative end tend to cluster more towards the blue end of the reference signature. And so what will happen is gene set enrichment analysis will calculate a running enrichment score. So every time gene set enrichment analysis gets a hit, the score will go up by a factor related to the t-score. Remember, the t-score is what's ranking these genes in the signature. And if I get a miss, then the score goes down. So eventually, gene set enrichment analysis, I'm going to call it GSEA for short because I'm tired of saying that. So GSEA will go ahead and calculate a maximum enrichment score. Any genes that contributed to reaching that maximum are considered leading edge genes. So in other words, those are the genes that we assume have the most biological relevance, and the ones I will be collecting for later meta-analysis across these different signatures. Okay? So from here, I've identified that I have 233 genes in the positive panel and 114 genes in the negative panel, the negative leading edge here. And now that I have these gene groups, I'm going to use these groups to identify, well, I've already identified, I'll talk about what they are in a second, verify them, and then of course compare them. Okay, I'm going to stop for a second because this is a load. You have to understand this. And I'll check for questions. So let's see if I have questions. Oh, I got questions. Cool. All right. So scrolling up, I've answered that one. Okay, question. What about microarray studies where one gene has been specifically knocked out to look at effect? Wait about 20 minutes and we will address that exact thing. And then you can re-ask that question, okay? So hold that thought. That's a really good question. Everyone says good work. Okay. Am I okay holding off that one question? Oh, here's another one. So these 347 genes you will be using for your work later on out of the hundreds, thousands, actually, tens of thousands technically. Yes. So these genes I'm going to have effectively in two different buckets. I don't want to mix the ends because then I can confuse them. So I'm going to keep my positives separate from my negatives, and we're going to keep them as buckets as we move forward. But yes, we will be keeping them together. Because that's how many samples came from the lab. I can only use what the lab gives me. So the lab gives me 3, that's what I take. Of course, if you want to go to the lab and generate more data for me, I'm all about that. Last call for questions here. Oh, you're welcome. You're all welcome. I love this. Okay, cool. All right, so I'll check back in the chat in a minute and we'll continue on. So what are these genes in my buckets? So from my positive panel, about 5.5% of the panel, 13 genes, were already reported in the literature as being associated with ICSARS infection. So this tells you that the approach of using gene set enrichment analysis helps us find some things that have already been identified. But quite a few of the genes we found have never been discussed in the literature before. So these are new findings that could be potential new targets. So to just take a general look, quite a few of them involve a zinc finger, and we are starting to see some data that zinc may play a role in how severe your COVID infection is. There's also several genes associated with solute carrier family, so things like electrolyte balance, sodium, potassium, that sort of thing. What does this mean biologically? I don't know, but it's something interesting as we move forward with this story. So we effectively did the same thing with pathways. Okay, so I'm going to take a signature, I'm going to take the positive end, the negative end, I'm going to define queries, I'm going to compare it using gene set enrichment analysis. I come up with pathway leading edges. Now, one critique from the gene study was, does it matter which signature you use for query and reference? We found with genes it does not matter. But when we did the pathways, we decided to be a little more robust. So instead of saying, yeah, it doesn't matter, we decided to do it and combine the results. So you'll see on one side, for example, we got 360, the other side got 359. Only 195 pathways were shared between those. So those were the ones we kept in our bucket. Doing the same thing with negative, we have 173 pathways, and we'll do the same thing. We'll talk about what those pathways are, we'll verify them, and we'll compare them to those deletion mutants someone was just asking about. Hold that thought. So what are those pathways? Well, again, about 9% of our panel, 17 upregulated pathways, were already associated with SARS infection. So we are seeing things people have already documented. But there are plenty of pathways not already documented. And quite a few of them are related to various immune system processes or viral response processes. Since we're dealing with a viral infection, it makes sense that you have upregulation of viral response pathways. I'm kind of surprised no one has talked about this before. Other interesting things we found, 19 pathways downregulated involved in ciliary action. So this is 10% of our downregulated pathways related to ciliary action. For those that don't know what cilia are in the lungs, it's the little tiny wave-like motion that helps you cough up phlegm. And we know that severe COVID patients really struggle to do that, and that ciliary action is diminished in those patients. Here's direct genetic evidence as to why. So pretty interesting findings here. So with that said, we wanted to know, can we reproduce these findings? two ways. One, using non-overlapping independent data sets, and two, using a random model. So let's take a look at verification. And at the end of verification, I'll stop for more questions. So we have our panels, and I'll start with genes and then I'll switch to pathways like I did before. So here's genes. So I have my gene panels that I've identified, and I have 4 verification signatures. One of them came with that super series. So it was made by the same people as my 2 identification signatures. So I'm calling this a true validation data set. The other 3 I found were made by other people. They were collected at the same time, but they'd use different cell type. So here they're using human adenocarcinoma lung cells rather than airway epithelial lung cells. Subtle difference, it's still a lung cell, but it might change my results, so they're not true verification data sets. So let's go ahead and take a look at the results. From my true verification data set, you will notice that my ICSARS positive panel clusters really nice. Positive panel at the red end, negative at the blue end. I get statistical significance. And when I do the random model, let me describe the random model for a second. Here I'm taking, what was it, 200 and some odd positive genes and 114, yeah. So I'm taking a number, 114 I'll say, random genes from this signature. And I'm rerunning the analysis to determine if the normalized enrichment score I get randomly is close to what I achieve from my actual panel. And so I do this 1,000 times, and you can see the distribution on the right-hand side here. And what's cool is that the NES, the normalized enrichment score from both the positive and the negative panel, is well outside what we got randomly. So this tells me the enrichment I observe from my actual panels is legit, is reproducible, is non-random. So it works. When I do the same thing with the 3 other verification signatures, the one in the adenocarcinoma cells, I see the same thing. Positive to positive, red end all the way down. The negative is the blue end all the way down. I get enrichment that's statistically significant. It's outside my random distribution. I'm really liking these gene panels. And to just look at a meta-analysis across these verification data sets, about 22% of the positive panel and 20% of the negative panel is shared in leading edges across all these panels. So that tells me that the genes in my bucket are coming up time and time again. And 10 of those genes already were specifically reported in the literature. So again, showing a good portion of what we're finding is accurate, but leaving room for new discovery. And there's my good old zinc finger again, gotta love that. So let's look at pathways. We're gonna do the same thing with pathways. So this top one is my true verification data set. The other 3 are my verification data sets in adenocarcinoma cells. What I want you to notice here is that the normalized enrichment scores are 10 and -10. Look at the range. I can't even fit these values on the random generated because the random generated barely made it to 3. So I am getting well outside of random generation. I'm loving pathway panels. And the same thing occurs where a good majority of my panels are reproducible across leading edges. And quite a few of them have known associations for immune response and viral defense. So again, confirming what we've already seen. Okay, so I'm going to stop here for a sec and I'm going to check for chats. someone wants to be head of the webinar to send a PDF. I'll let administration take care of that if they want to. Any other questions, comments? It gives me a second to drink my water. Okay, so let's continue on and get to that question about the knockout models, the deletion models. So my aim 3 is to compare my buckets to other strains and other models. So I'm going to start here with gene expression, and as before, I'll switch to pathway. So first, I'm going to take the ICSARS panels that we found in Petri dish, and I'm going to compare it to an ICSARS infected mouse and a mock mouse. And I'm going to do the same thing. Now, what's interesting when you're comparing the human to the mouse model is that the positive panel looks really well. It aligns with my red end. I get statistical significance. It's within 2 of my 1,000 generated random trials, which is still outside statistical significance. So the positive panel holds. The negative panel, though, does not. So you'll see a non-random placement. You'll see a NES that's close to 0. My p-value is definitely greater than 0.05. So it looks like my negative panel of genes does not transfer into the mouse model. So I have to think that it's also not going to transfer into the actual human patient. But the positive panel might. So something to keep in mind. Now, to look at the genes that are shared between the human Petri dish and the mouse biopsy samples, I do this Venn diagram, which can be a little confusing. So I'll stop after this, well, actually after the next slide to ask questions. So out of all the genes that we found shared in the identification verification signatures, 9 of them overlap with the leading edge from the mouse model. However, some genes were not included in the mouse model, but were in all of the human platforms and shared in the leading edges. And some genes were not included in either of them. So it wasn't fair to throw those genes out just because someone in the lab didn't look at them, or maybe the mouse doesn't have them. So we kept those genes in just for safety. This gives us 20 genes identified with iCSARS, both in human and in mouse model. Here are those 20 genes, and here is their T-score across all the signatures. The top 2 are identification, the next 4 are the verification, and the last one is the one from mouse. And again, the black squares show you which genes were not measured as part of which signatures. But all of these then have been identified by GSEA, gene set enrichment analysis, as potential candidates for targeting an ICSARS infection. Okay. So I'm going to stop here for a second. We will get to those knockout models, just hold tight. Any questions? What computational tools do you use for gene set or for gene expression analysis and pathway analysis? So I don't know if you're coming in late or not. All of what I'm doing here is GSEA, which is downloadable from Broad Institute. I personally prefer the desktop Java version because I could never get the Python version to work, but GSEA is also well in R. So if you want to use R, you can do that also. Pathway analysis is GSEA with gene signatures and the gene ontology thing. So, okay, someone's going to be sharing that. Cool. All right. Any other questions? Okay, then. So now let's look at those knockout. Why did they use? I have no clue because I didn't conduct the experiment. There are some monkey models that are popping up for SARS-2, but we're not using them. Can you use Enricher? That I'm not sure. That I believe uses a Fisher's exact test, not gene set enrichment analysis. So it would not be as sensitive as what I'm using here. It's not perfect for this. I agree with your assessment there, Sudip. Any other questions, comments? Okay. So then let's go back to this. So now that I know those 20 genes are associated with ICSARS infection between my models, I'm going to look at other strains. And I'm going to start with the SARS-1, then I'm going to move to MERS and SARS-2. So I have other virulent strains. Urbani is actually a derivative, or I should say the parent, for ICSARS. And so I have one signature in human. MA15 is mouse-derived Urbani, and I have 8 signatures with varying inoculation doses. And then here are those knockouts. So there are several attenuated strains with different genes knocked out in order to make the virus less virulent, less pathogenic. The gene itself will not matter, but these were all originally developed to come up with a vaccine for SARS-1 that we never did because we ran out of funding. So just be aware of that. So this is a pretty busy slide, but you'll notice all the gene signatures that we looked at. We looked at them across human models, across mouse models, literally any data set I could get my hands on that harvested at 48 hours, I was on it. You'll notice that the positive panel holds for all of them. Statistically significant too. The negative, not so much, but that wasn't a surprise because we saw that with the ICSARS mouse model. On the right side, you're going to see random model. The only positive panel that did not pass random model was this one right here, and that was the lowest inoculation dose at 10 to the second power. Okay, so there is a detection limitation to our approach. So if we have such a light infection, we might not be able to pick up these genes transitioning or changing within the cell. So we removed this one signature from further analysis and we kept everybody else. I see the question, but I'll get to the question in a second. So we did the meta-analysis thing with the Venn diagrams across all the different SARS-1 strains, excuse me, and we came up with 5 overlapping genes. Out of those 5, here's the heat map. You can see which signatures are included in what genes. And we're seeing these 5 genes are actually related to interferon. They're part of the interferon pathway. So that's pretty interesting that 5 interferon genes are shared across all those different strains of SARS-CoV-1. Now I'll get to MERS and 2 in just a second, but I'll stop for questions because I did see that question. Is there any pre-ancestor originally, not that I'm aware of, at least not that I used in this study. There are other beta coronaviruses, but they weren't life-threatening, ergo no one ever did research on them. Next question, how many mutations are you seeing in the SARS-CoV-2 strain? This I don't know because they didn't do the genetic analysis on them. This data is so new, the papers are still all in peer review. And so I'm not getting that, I'm just getting the data. And the duration of the mutation, like a month, I'm not sure on that either. And we're not to SARS-2 yet, so quit jumping ahead. Okay, so that's that. So let's get to SARS-2. So here's MERS and SARS-2. Same idea. You can see the positive panel does really well. In this case, the negative panel does pretty good too. This bottom one though, this GSE 155518, I actually asked the people who made this data set because it was such an odd finding to get statistical significance in the reverse. And they said that they withdrew that data from consideration in peer review because the RNA didn't come out as clean as the other data set they made that was right above it that did work. And so that's interesting that how we extract this DNA does seem to matter in terms of the approach's ability to detect significance and genes and that sort of thing. Yeah, it's interesting. They're part of the cytokine storm. Keep that thought because we haven't got to pathways yet. You want to see cytokine storm. Again, random modeling, things look good for the positive panel, but not as much for the negative. So you get the idea. So my I combined then between my 5 SARS-1, MERS, and SARS-CoV-2. The same 5 pop out. Here's that heat map. So those 5 genes seem to be consistently identified across 37 signatures and all these different SARS strains. Okay, so I'm going to take a moment, check the chat. Which gene is mainly responsible for the gene-causing ability of SARS? I believe personally they work together. I'll show you why in just a second. Okay, so let's get to that cytokine storm. Let's talk about pathways. So the same idea, I'm just not breaking it up as succinctly because I'm running out of time, definitely running out of time. So here you see the positive panel looks really good for pathways. And look, even pathways overcame the limitations. So I'm getting statistical significance, whereas here's my -2. And if I look across, it passes random model. And then for this one at the bottom where we had issues with the RNA being extracted, it barely passes, but now it's passing. So it looks like the pathway activity approach may be better at detecting some of these changes than the gene expression approach is. Something to think about. Do the whole Venn diagram thing, come up with 11 pathways that are shared across all 37 signatures and all these different strains and models. So what are those 11 pathways? Well, in this case, because we could have a complete data set, I was creating pathway signatures from gene ontology. I can rank these pathways. Notice we're not missing any data here. And so by ranking the pathways using a Stouffer z-score, type 1 interferon becomes the top pathway. And what's interesting is out of the 5 interferon genes I showed earlier, 4 of them are in this pathway. So 4 of those 5 genes are really driving this pathway to be the top. And I'll talk about interferon clinically in a minute. But let's pay attention to the other 10 pathways that were identified here. Because notice, negative regulation of cytokines, CD4 positive cells, these are T helper cells, T activation, T differentiation, type 2 response, which is an adaptive immune response of which T helper cells are involved in. So you see all of the pathways that are found in addition to interferon are all related to a very strong T cell immune response, particularly focusing on the helper cells. And this has not been reported in the literature specifically, but a lot of clinical evidence shows increase in cytokine storm, increase in immune response, etc. So it's not a surprise to see these pathways. More work does need to be done to see which genes in these pathways are really driving the pathways, because we didn't see that with the genes. But that's for another talk. See you in the fall. My work is never done. All right, so I'll go through conclusions and then I'll take questions at the end. I do see the chats going. So my overall goal here was to figure out molecular changes associated with SARS infection through using a computational analysis of mRNA expression data with the idea that those genes and pathways could be potential therapeutic targets. And to that, I identified genes and pathways associated with SARS-1 infection in a lung human Petri dish culture. Some of those genes were identified prior, some of those pathways were identified prior, and some genes and pathways were not identified prior. Then I verified those genes and pathways in other SARS-1 infected human lung cell cultures. And so reproduced them with those non-overlapping data sets, found reproducible genes, some of which we've talked about, some of which haven't been talked about before. Then we took those gene and pathway buckets and we whittled them down to the most important by looking at mouse model and other strains. And we found those 5 overexpressed interferon genes that are really driving a type 1 interferon pathway upregulation, along with 10 pathways related to T cell activation and T cell helper. Okay. So before I take questions, the number one question I would be asking is, can we use interferon therapy? Has this been done before? Give me clinical data. So I went and looked. And yes, it's being used in clinical trials, even in India, which is super cool. Now, the trick with interferon therapy right now is that we don't know how to use it best. Do we use interferon alpha, beta, gamma? Do we mix it with another antiviral drug? Do we give it early or do we give it later? That all is being discussed through several reports. And that information is unclear. Do we give it as a shot? Do we give it as a pill? All of it is up for debate. But whenever we look at pre-treating cells with interferon beta, that was protective against them getting COVID-19, SARS-2 infected. So, you know, there is evidence already that is showing interferon therapy may be very promising. And so I'm super excited that my computational study was able to really pick this up and try to move this forward. Okay. So I've got to thank my people. I actually did this over 2 universities. I originally started at Davenport University in Grand Rapids, Michigan, and my lab there was called the Harris Interdisciplinary Research. And so Amber Park is a recent graduate from there. She's the co-author on my SARS paper, both of them actually. And then Marius is a MSU student who came on board when I switched to MSU, and he's a co-author on the pathway paper. Okay. And now I'll take questions. So I'll head back. I've seen a ton of them in the chat. So let's scroll back a little bit. Okay. When and where will the link be shared for the certificate? I'll let someone else get this. Which microorganisms mutate fast? All right, so the way that works is what we call generation time. The faster you are able to reproduce offspring, make babies, the faster you will be able to mutate. So viruses inherently are faster than bacteria, who are faster than fungi, and they're faster than us. At least we beat dinosaurs, I think, maybe elephants, but you get the idea. So yeah, we're really worried about SARS-3 coming up. My guess is the next 5 years, but I hope I'm wrong. How do we come to know that COVID virus comes under SARS group? So that was done really early on using genetic sequencing and phylogenetic tree. We have found that SARS 1 and 2 are like 80% identical in in RNA structure, and they're only 60% to MERS. So MERS is kind of a cousin, so to speak. Why virus is so adapted that the fastest mutation, especially in COVID virus, all viruses mutate fast. It's not specific to COVID. Can I explain about Delta plus variant? Stay away from it. That's my explanation. It's one of the strains emerging that will give us SARS-3. So it's just going to be the way that works. You need to go for metagenomic analysis. We'll find similarities in groups. I agree. There's networking and other things I agree need to be done. Not exactly clear what ICSARS is. So ICSARS is infectious clone SARS. So what happened was they captured Urbani from nature, and then they put it into a lab petri dish. And the passage of the wild type to a lab strain is why they changed the name. So effectively, ICSARS is the same as Urbani. The only difference is Urbani is catchable and ICSARS should not be. Maybe I missed, we did not see any ACE2 expression changes, but then we looked at a gene level and didn't see it, and I didn't see anything with it in a pathway that came up. We did see some cardiac genes come up, so there might be something in there to look into. Instead of using vaccines, why not use interferon therapy? That's a good question. More research would need to be done. It may be easier than trying to like freeze and ship some of these mRNA vaccines. That's a real pain in the butt. Okay, let's see. Which genes are responsible for viral receptors? Yeah, none of these are viral receptor recognition proteins. These are all inflammation response. But in theory, if you lower the inflammation response, you then can get, people that are more likely to survive. Genetic changes made mostly by introns and exons. We did not look at any sort of alternative splicing, did not look at genetic drifts of anything. Okay, I'm seeing, oh wait, here's another one. Interferon storm is associated with vitamin B and zinc deficiency. Oh shoot, it just moved on me. Can upregulate it. That would be an interesting idea. I did not run any vitamin D or zinc deficiency or zinc pathways because they're not available in gene ontology. However, if those become available, that would be a real interesting thing to see. I'll agree with that. Okay, cool. Nanotechnology useful. All right, so we're going to try to stay on topic. Do I have any other questions I may have missed? There's a ton of thank yous. I appreciate that. It's been a long year for us all. Here someone's asking about the variations in vaccines. To be 100% sure, I am 100% honest, I am not sure about vaccines outside the United States because I only work with the American government. I am not high enough on my totem pole yet to work with the world government, but my boss at ASM is. So I could ask him. As far as me personally, I got the Pfizer vaccine. I have seen good data from Monera. Johnson & Johnson, I mean, it's better than nothing. I've heard decent things about your COVID shield, but I've heard mixed results also. But I'm the wrong person to ask. Have I faced complications during this? Yes. I would say one of the biggest complications was with the SARS-2 because of the fact that that was not published and the stuff was so new. So I really am not clear on the strains they used for those studies. I'm not even sure they're clear on the strains they used for those studies. And we're constantly getting nitpicked about that in the peer review. People keep wanting us to update for the most recent SARS-2. And that's fine for a while, but seriously, I need to publish this sooner or later. So, you know. Yeah, I don't know anything about the COVID associated. That's your mold. That's that black fungus stuff that you've been growing. I've read about it. I don't know much about it. I haven't seen any data on it, but I would like to look at it. I'll give you that. That is intriguing to me. Someone's asking me about LinkedIn. And that's okay. I can repost that wherever that went into the chat box. Okay, here's my LinkedIn. And I also have my email address here. You can email me at ostry@msu.edu. I'm also at training@msu.edu. Both emails go to me. Oh, wait, somehow I got into someone's private. Oops, sorry, hold on. Try to make this public. Nope, still private.
What about the so many variants arises from SARS-CoV? In what way you are deviated from your studies?
Can you repeat your question, please?
So many variant arise in SARS-CoV-2. So in what way you overcome to this topic, gene expression analysis? Particularly, you take this genes for your study now. So how can you?
Okay, so one thing in theory, based on what we've seen, interferon should hold regardless of what SARS-2 strain you're looking at. Assuming, of course, you've got a strong enough case of it and you've got, you've extracted the RNA well enough. The trick, though, will become trying to tease out any variation for strains that are out there. And I don't think that's going to affect our data that much here because of the fact that we looked at so many different strains and it keeps reproducing. Does that make sense and answer your question?
My question is, the variant is mutated. Due to mutated, the variants arises. So is it affected? Is it make your data is you took 300 genes for negative, 300 genes for positive? Is the negative thing is a variant mutated one?
The variants we used did have known and unknown mutations. So for the SARS-1, we knew what those mutations were. For the SARS-2, we did not know what those mutations were. And I would assume that if we added data to it, even if we did or did not know the specific genotype of the variant, we should still see these results.
Okay.
I'm not sure I completely understand your question enough to answer it.
Yeah, okay. I can study more regarding your paper. Then only I can again mail you regarding my questions.
Okay.
Thank you. Nice.
Yeah. Thank you. I'm trying to get the link in here, but it keeps only making it private. Is there a probability of another vaccine? Yes. Pfizer actually have a second booster shot for the UK B117, I think it is, variant, and a couple other ones. So yeah, we are expecting booster vaccines. The question we were wondering is how long it's going to take until we need the vaccine booster. So far, it seems like we can do this with the annual flu shot. So once a year, you would get your booster shot. We really don't want to do it less than once a year. And if we could get by with more than once a year, you know, like every 2 years, that'd be best. But I don't think that's feasible. Opinions on efficacy of nasal vaccines. I have not seen much data on them. My only concern with that is you have to cross the mucous membrane barrier in the nose, and therefore you're going to require a much higher dose than if you were giving it into an arm, you know, an IV injection. So I personally would rather get an IV, but I can understand some cases, perhaps pregnancy. We do this with pregnancy, where anyone that's immunocompromised, we will give the nasal injection for the flu shot. So I could see some place for it, but me personally, if I can take the shot, I'd rather take the shot. Is there any variations genes are present that I don't know? Can we get clear-cut idea between strains and variants? Well, we have some idea. So like, I'll go back to my list. Where'd my list go? This guy. So we know, for example, that the bat SRBD strain is the virus that infects bats, but we put a human ACE receptor on it, and that's how we're able to get it to infect humans and mice. That's the only difference. For these other ones, like the Delta ORF6, so this is a point mutation in that one gene. Everything else about it is the same to your Urbani or your ICSARS. But that one point mutation in that one gene makes that strain less virulent. So we knew what we were doing when we targeted these genes, and we know what we did to target these genes. These are all man-made strains, so to speak. These MERS and SARS too, though, are not man-made, and so I don't know anything about them that way. Does the child of pregnant woman get infected with pregnant women get vaccinated? That I don't know. I know the data's out there, I just don't remember it. I want to say I read a report that there is a short-term immunity, but then I don't even remember if that fades or continues as a result of breastfeeding. So I'm not sure, but I know it exists. So ask the Google overlord. Any other questions?
Dear participants,
Yeah.
if you still have any question, you can post it in the chat box. I will also be sending the feedback link. But before that, now I would request our founder coordinator, Aryadeep Das sir, to kindly grace the participants. Over to you, sir.
Okay, thank you, Sneha. So a very good evening to one and all, and very good morning to Dr. Harris. So my heartfelt thanks to our guest speaker, Dr. Laura Harris, and I thank you for accepting my invitation for the webinar. It was an academic first, and the topic of your research is so relevant and useful in this time. And you know, the way you shared your knowledge regarding this topic was so impressive and encouraging. Thank you for making the time to share your work with us. And a big thank you for answering the questions of all these body researchers. Thank you, Dr. Harris. So it was really indeed an awesome session, and it was interactive session. So, and also my heartfelt thanks to our organizer, event coordinator, Dr. Ritu Chandel, Dr. Sujit Das, Dr. Arpana Sharma, and our coordinator, Ms. Sneha Mahajan, Ms. Ananya Chakrabarti, Mr. Shreyans, and our faculty members, Ms. Binita Tiwari, Ms. Somuju Bhargavi, Ms. Priyanka, and Dr. Kavita, Dr. Fatima Tasriwala, for inviting Dr. Amod Kumar. And my dear students and researchers, thank you for joining and invest your precious time for listening the talk and support us. Now, but not but least, on the behalf of Biosetup Lifescience, I thank you each and every for their contribution to make this webinar a successful one. Thank you, everyone. Thank you. And now I request Dr. Sudip Das kindly take opportunity and give a vote of thanks. Thank you. Thank you, Dr. Harris.
Thank you so much, sir.
Thanks, everyone. I'll still hang here. I see a couple questions.
Yeah, we will looking forward for many events. It was really indeed an awesome session.
Thanks. I had fun sharing. I look forward to August.
Yeah, sure, definitely. Thank you.
Thank you so much, sir. Now I request Dr. Sudeep Das to propose the vote of thanks. Over to you, sir.
Hello, everyone. Hi, I'm Sudeep. Yeah, I mean, I guess Orgadeep has already thanked everyone who was relevant. I would just like to take this opportunity and say that it's really a delightful experience and really informative. And Dr. Harris, thanks a lot for sharing your research and very interesting analysis. Yeah, also quite thought-inducing, as I also had a couple of questions and quite relevant as we discussed this before. Yeah, and I think I would say that this is for me the first webinar from Biosetup, and I think they're doing a great job. I think Orgadeep, I want to really say that Orgadeep is really the pivotal person in pulling all these up and creating these fantastic workshops and webinars. And so I would like to thank everyone who has been here. I hope you all learned a lot from Dr. Harris's research and take home important information. And I think you guys had a great discussion. The chat was really lively and a lot of different questions from different sort of different areas of SARS-CoV-2 infection and COVID-19 in general. And yeah, and I would really I'm really grateful to all the organizers for inviting me. And I'm really grateful to Dr. Harris for sharing her research. And I hope that in the future we continue exploring different avenues of making different webinars, seminars, and workshops. And all the very best to Biosetup Lifesciences for their future projects. Thanks a lot.
Thank you so much, sir. Dear participants, you can directly mail it to ma'am if you have any questions. Ma'am has shared her email ID also. And the feedback link will also be shared in the WhatsApp group. And you will get all the recordings and the PDFs which you were asking. We will be posting it in the group. A big thanks to everyone for joining with us. Thank you so much, ma'am. Thank you, ma'am. And thank you, everyone.
Thanks, everyone. Have a great evening.